Hello, and welcome to the Geological Society's second virtual public lecture of the year. So due to the ongoing situation with COVID-19, we've converted these public lectures into live virtual lectures. So it's actually a pleasure to see some of you here in this new forum. So throughout this lecture, your microphone will be muted and your video cameras will be switched off. So you will be only able to hear and see the speakers. So my name is Alexander Whitaker. I'm a senior lecturer in tectonics at Imperial College London. But additionally, I am chair of the science committee at the Geological Society. And I'm delighted to host the event today. So throughout 2020, the Society has been marking the year of life through a series of conferences, lectures, and educational events and resources. Today's lecture will explore animals with backbones, vertebrates, that have an evolutionary history of nearly 500 million years, with fossils instrumental to understanding how the group came to be so hugely successful. So today's speaker is Sam Giles, Royal Society Research Fellow at the University of Birmingham. Prior to this, she completed her MSci in Geology at the University of Bristol in 2011, and a DPhil in Paleobiology at the University of Oxford in 2015. She then spent 2 years as a junior research fellow at Christchurch, Oxford. So Sam's main research interests are in the relationships and morphology of the bony vertebrates. So she uses non-destructive X-ray imaging, CT scanning, to unlock the external and internal anatomy of living and fossil vertebrates. She particularly focuses on the early history of bony fishes, which account for 99% of all living vertebrates. Her work provides insight into the origins and evolutionary success of different vertebrate groups. So, at the end of Sam's talk, there will be a 15-minute question and answer session. So, if you have a question that you want to get answered, please submit that using the Q&A box function within this Zoom webinar. We're going to try and get through as many of the questions as we can, but it's possible that we might not be able to get through all of them in the time allotted. So please accept my apologies if we are unable to get to your one particularly. All right. Well, without further ado, I would invite Sam to turn on her microphone and camera and start to introduce today's public lecture. Sam Giles, thanks for joining us.
Hi, everyone. Thanks for coming along today. So I'm going to be talking to you today about vertebrates. Vertebrates, animals with backbones, are an amazingly diverse and successful modern group. There are key features of their body plan that allow them to occupy almost in any environment you can think of, from the land to the deep sea and skies to deep underground. But there are major obstacles to understanding how this hugely successful modern group evolved. Firstly, their evolutionary roots stretch back some 500 million years. And secondly, many of the most informative aspects of their anatomy are internal and therefore hard to access. So my research uses 3D X-ray imaging techniques to remove these barriers and get inside the heads of early vertebrates. Doing so allows me to answer fundamental questions concerning the origin and evolution of vertebrates and investigate how life has responded to past environmental crises. I'd like to end with my conclusions. So here are my acknowledgements at the start. I'd like to thank these institutions on the left for funding my work and supporting my research, and to the people on the right for collaborations, access to specimens, and very helpful discussions. Almost all living vertebrates fall into two groups. So we have the cartilaginous fishes in purple. These are sharks, rays, and chimeras, and there are around 1,000 living species of them. And then the bony fishes. The bony fishes have a vast modern diversity of some 60,000 living species, which fall nearly equally into two groups. We have the ray-finned fishes in green, which include things that you'd see in an aquarium or on a dinner plate, so things like trout, seahorses, and flatfish. And then the lobe-finned fishes in blue, which include fish like coelacanths and lungfish, but also all land-dwelling vertebrates, such as frogs, lizards, birds, and mammals, including ourselves. My research encompasses all bony fishes and indeed other vertebrate groups, but with a particular focus on the ray-finned fishes. And in particular, I want to better understand the relationship between living and extinct groups of vertebrates, and use this framework to answer questions concerning the origin of major vertebrates, of major features of the skeleton, in order to constrain the appearance of key innovations. The relationships between fossils and living taxa, and trying to understand when living lineages evolved from a haze of fossils. And to identify how ray-finned fishes survived and were shaped by the many environmental perturbations that have punctuated their history. We can try and understand the relationships between living groups by putting them into an evolutionary tree. And molecular evidence where we look at DNA tells us that the different vertebrate groups have these relationships. So ray-finned fishes and lobe-finned fishes are most closely related to each other, and they're united as the bony fishes. And then cartilaginous fishes are the closest living relatives to the bony fishes. And together, these three groups make up the jawed vertebrates. And these groups are named for their skeletons. So we have the bony and cartilaginous fishes, and this will be important for a bit later on in the talk. The fossil evidence tells us that the two main groups of vertebrates with jaws, so the cartilaginous fishes and the bony fishes, separated over 425 million years ago in a geological time period called the Silurian. This means that fossils are incredibly important for understanding how different groups of early vertebrates are related to each other. We only look at living animals, we're just seeing a tiny snapshot of the whole history of these groups. But this is where we run into the first problem. I've already explained how we look at relationships in living groups, normally by using molecular data. So we can take gene sequences and work out which genes are shared in common with different groups and which are therefore most closely related to each other. But what happens when we add in fossils? So here we've added in a placoderm on the left, this animal here, and an acanthodien. Placoderms were some of the first animals to evolve jaws and teeth, and acanthodians are kind of spiny, they're known as spiny sharks often. Molecular data isn't available for these groups, despite what things like Jurassic World would have you believe. So we build our understanding of relationships by observing the anatomy of each of the animals that we're interested in and breaking this down into different characters. Then we assess the distribution of those characters across a larger group of taxa. So for example, we could ask whether a limb, such as our arm, which has a single articulation point with a shoulder, is present. We have our list of taxa, and we assemble our morphological data and assess the state of this character across all of our animals. So in these examples here, we're looking at the number of contact points between the shoulder girdle and what kind of attaches to it. So in the placoderm, the shark, the acanthodian, and these two ray-finned fishes, you can see that there are multiple bones which contact the shoulder girdle. So we'd say that these things don't have limbs, they're absent, they have fins instead. But if we look at the coelacanth and orangutan, they both have one bone contacting the shoulder girdle, which is exactly the same as our limb. We have our upper arm, which connects to our shoulder girdle. So we'd say that this character is present in these two animals. And in fact, that's one of the characters that supports a close relationship between things like primates and coelacanths, to the exclusion of everything else on this screen. In fact, we're united in a group called the lobe-finned fishes. And this process of identifying characters is repeated for hundreds of taxa and hundreds of characters. And we build up this data matrix where we assess the presence and absence of different states of all of these features. We then run that data matrix, we analyze it in a quantitative framework, and that allows us to build up a comprehensive picture of relationships across lots of different groups. In particular, we can use this to work out how living animals are related to fossil animals. And we can build up this tree of relationships where we see that acanthodians are quite closely related to cartilaginous fishes. And then the placoderms kind of diverge, they branch off before cartilaginous fishes and lobe-finned fishes diverged. But this is where we run into our second problem. Our understanding of the relationships and the evolution of different groups is hugely biased. In particular, we know a lot more about the lobe-finned fishes. And this is because they lead to our group and eventually to ourselves. So on the left here, this is a diagram called Geological Tree of Life. This is from the 1860s. And it shows this very kind of old-fashioned view of evolution, where you have different kinds of animals and amoebas and things like that, eventually leading to man at the top, this kind of very old-fashioned kind of view of humans being this pinnacle of evolution. But as a result, lots of attention has been paid to early members of the lobe-finned fish group because they chart our own evolution. So for example, the evolution of fins into limbs. But ray-finned fishes, in contrast, have been quite sorely neglected, despite the fact that they contain over half of all living vertebrate species and are every bit as diverse and disparate as the lobe-finned fishes. Many of the major innovations that drove vertebrate success are tied up in the braincase, and this is a kind of bony box that sits inside the skull and houses the brain and sensory organs. It contains key information about how the animal would have behaved and functions, and because it's morphologically very complex, we can use it. It's really important to use to build up a kind of picture of relationships between different groups. But here we run into the third problem. It's an internal structure. So here we have the skull of a sturgeon, and we have the external bones of the skull roof in grey, and then the internal skeleton, the braincase and vertebral column on the left here, on the right here. So it's an internal structure. It's hidden by skin, muscles, and the external skeleton. And in the case of fossils, it's also hidden by rock. So how do we get at it? Well, historically, people would get at the inside of fossils using something called Solis's grinding machine. And this has been used since the early 1900s. This is a kind of terrifying looking machine where you put your fossil in a chunk of plastic, you attach it to this grinding plate, and then you turn this handle and you grind away a small amount of the fossil. So anything kind of less than a millimeter. Once that's done, you take your fossil off the plate, you draw a picture of it, you put it back on, you turn the handle again, and grind off another millimeter of rock. And you repeat this process until you end up with a stack of drawings, which you then cast into wax plates and use to build up a 3D model. And then you also have a pile of dust where your fossil used to be. So unfortunately, museum curators don't really like you grinding their precious specimens into dust. So I use CT scanning instead, where I use X-rays to unlock the internal anatomy instead of destructive sampling. And because different materials attenuate X-rays to different amounts, we can exploit this to understand the internal and external structure of the object of interest without having to cut into it. This uses exactly the same principle as getting an X-ray at the hospital. So your bones attenuate X-rays a lot more than your skin does. And that means essentially that you can look through your skin and your muscle and you can see the bones within. And we load these graphics into a specialized program, and we can cut through the fossil to look at the internal anatomy. So here on the top left is a photo of a fossil fish skull, just as a guide. We've got an eye showing you the position of the orbit and some teeth in the jaw. And then the vertical white line is essentially a section where we've virtually chopped through that fossil. And different materials show up as different gray values. So all of this black around here is the air. This kind of gray material here is the rock. And then this darker gray material is the bone. And we can digitally dissect out each of these bones and build up a 3D model of these different objects. So here's our example. We start with the fossil on the left, then we do a section through it to look at the different materials inside. And we can dissect out each bone and eventually look at individual bits of the anatomy, including on the far right here, the endocast, a kind of infill of the cranial cavity that gives an approximation of what the brain would have looked like. And these models can be manipulated and even 3D printed. And they allow us to look at the anatomy in really high amounts of detail and feed kind of new data into these very old problems and investigate some really long-held theories and see whether they hold up. So I've shown you how these amazing techniques work. And what I want to talk about now is how I apply them to my research. So as I said, I look at early vertebrate evolution, in particular the relationships between different fossil groups through time, and trying to understand how those fossils fit in with the living animals. And from this, I can understand character evolution and major innovations. And the first question I want to consider is the origin of jawed vertebrates, so the point at which the bony vertebrates and the cartilaginous vertebrates separated from each other. Here we have the diagram that we saw earlier with cartilaginous fishes and the bony fishes separating from a shared common ancestor over 420 million years ago. And because sharks stayed in the water and there are fewer species of them, there are only around 1,000 species, whereas bony fishes became much more diverse in terms of number of species, they kind of went onto the land and eventually led to ourselves. There's a tendency often to view cartilaginous fishes as somehow primitive or less highly evolved than bony fish. And tied up with this is an assumption that they retain this kind of ancestral morphotype. So that is, they look like the ancestors of what both groups would have looked like originally, and they haven't really evolved since that time. And this seems to be held up, at least superficially, by some aspects of their anatomy. So sharks don't have bones. Their skeletons are made up of cartilage. But bony fishes have both bone and cartilage in their skeleton. And you can see this by looking at your own skeleton. So if you think about the kind of bones that make up your arm or most of your skull bones, these are a special kind of bone that's got this kind of spongy texture in the middle. So if you think about the bone marrow in your long bones, that's this kind of spongy texture. We do also have some cartilage. So if you give your ear a wiggle or the end of your nose, you'll feel something that's quite firm, but also very, very flexible. So the entire skeleton of cartilaginous fishes is made up of this cartilage, whereas bony fishes have bone and cartilage. And when we put fossils into the picture, this story seems to be held up. So we've got these things called Acanthodians. As you remember, these are related to cartilaginous fishes. These don't have bone either. And then it looks as though placoderms also don't have any bone. And bone seems to be the first to, bony fish seem to be the first to evolve bone, with cartilaginous fish lacking bone. So my colleague Martin Brazeau at Imperial College London has been interested in questions like this for a while. So he ran several field seasons in Western Mongolia to look for new early Devonian taxa. And this is a really critical time period in the evolution of jawed vertebrates. I was lucky enough to be able to attend one year. So here are some obligatory embarrassing photos of me looking at fossils in doing field work. And we found a really cool location in the far west of the country, which had got some really interesting looking fossils, but they're very challenging to work with. So down here in the bottom right of the screen is one example. And you can see that the fossil is actually preserved as a mold. So rather than it being a kind of plate of bone on the surface of the rock, there's a cavity within the bone. And that's what contains all the morphological information. So I'm going to talk about one particular specimen. Here on the left is what I'm going to say is a really interesting fossil from Western Mongolia that's 405 million years old. Now I say it's a really interesting specimen, but it looks pretty awful. And that's because the fossil is preserved as a mold. So all the important stuff is hidden in a cavity inside the rock. Luckily, this is where CT scanning is really useful. And when we CT scanned this block and made a 3D model of that cavity, we found that it's the skull of a kind of placoderm, so one of these kind of first vertebrates to evolve jaws and teeth. And specifically, it contains the skull roof in blue and kind of silver, and the braincase in this kind of goldy brown color. And we can make a 3D model of what the anatomy looked like, and we can spin it around and look at it from different angles and use this to work out what kind of animal it would have been. We can also fill in the cavity within the braincase, the brain cavity. This is in purple, as you can see now. And we can use this to get an idea of what the brain of this animal would have looked like. We can even kind of reconstruct individual nerves and blood vessels of this animal. To orient you a bit, we're just looking at the skull. Here's the kind of the top of the skull and the bottom of the skull. At the top here is the anterior, so where the nose would have been, and at the bottom is the posterior, so where the kind of the neck of the animal would have been. We've got some kind of googly eyes showing you the rough position of the orbits. So when we try and work out where in our evolutionary tree this animal goes, it comes out here. It comes out kind of near other placoderms and before the cartilaginous and bony fishes separated from each other. But what's really unexpected about this animal is when we look at the tissues that make up its skeleton. So because of the position where this animal comes out in the tree, we'd expect it to have a skeleton made of cartilage with no bone. But that's not what we see when we look at some sections through the skeleton. This animal's skeleton seems to be very much made of bone, so it's a bit difficult to work out, but you can see this kind of spongy texture, very different from the cartilage that we would expect to see. And when we compare it to a bony fish here on the right and a cartilaginous fish here in the middle, you can see that, as I just said, the structure is very, very similar with this kind of spongy texture to what you see in the bony fishes. And why is this important? Well, it has major implications for how we think that bone and skeletons evolved. Rather than cartilage being primitive and bone only evolving in bony fishes, it looks very much like bone evolved a lot earlier than thought. Rather than it being a special feature of bony fishes, it seems like it first evolved in some placoderms, like our Mongolian example here. And then rather than cartilaginous fishes being primitive for not having bone in the first place, it seems like they're specialized because they secondarily lost it. So at this point in kind of the early evolutionary history of cartilaginous fishes, the early members of the group lost this feature. And there are many reasons why that could be. It could be that bone is kind of nutritionally very expensive to produce, so they decided that they were better off without it. It's also very heavy. Perhaps it suited the early the early cartilaginous fishes to not have that skeleton. But rather than cartilaginous fishes being this kind of relic that hasn't evolved since it first appeared 425 million years ago, this feeds into this picture that cartilaginous fishes are actually very specialized, and they're every bit as specialized as the bony fishes that we see as well. So far I've been talking about the origin of the jawed vertebrate, so the point at which the cartilaginous and bony fishes separated. And next up, I want to talk a bit about how we integrate the living and fossil fish records to try and understand when the living groups of animals evolved. For the rest of the talk, I'm going to be focusing in on just the ray-finned fishes here, and we can have a look at their relationships a bit more closely. So the relationships between different groups of living ray-finned fishes are fairly well known, and they're quite well supported by both molecular and morphological data. There are a number of living groups, but there are two important names that we're going to talk about, and I want to specifically mention. So Polypterids, which is this group here, and there are around 16 species of them, 12 species, sorry. And then the Actinopterans, which includes everything else. So from paddlefish to bowfin to gars and also teleosts. As I said, there's a huge imbalance in species richness. So the vast majority of ray-finned fishes, nearly 36,000 species, are actinopterans, and specifically teleosts. There are only 12 species, living species of Polypterids. And this has led to non-teleost ray-finned fishes often being neglected in debates about relationships. But seeing as teleosts evolved significantly after all ray-finned fishes, it means a huge chunk of ray-finned fish history is being neglected. And the earliest ray-finned fishes, particularly those from the Paleozoic, so over 250 million years old, have been particularly poorly studied. And this makes it difficult to know when the different living groups evolved. The split between the living groups is known as the crown node. So this is the origin of the living radiation. And as I said, we have quite a poor handle on this. But this is a problem because we need to have an accurate idea of when different groups evolve so that we have a robust timescale of evolution. And this is necessary for understanding everything from rates of gene mutation to rates of extinction and diversification. So how do we get at the age of the crown node? There are two main ways. We can use rocks, looking at the fossils, and we can use clocks, looking at molecular data. So the first one that I mentioned is the first way of dating the crown node is to use rocks, to use fossils. So we look at existing hypotheses of relationships, and we pick out the oldest fossil that is believed to be related to a living radiation and fall within the crown node as a result. And the oldest fossil thought to be within the living radiation is about 380 million years old, 384 million years old, and it's from the Devonian of Australia. So this indicates that the living groups must have separated from each other at least 384 million years ago. But if we look at molecular clock data, we get a much wider range. So estimates anywhere from around 300 million years ago to well over 430 million years ago. And there are multiple factors to explain this incongruity and why we have such a broad range of dates and such a mismatch between the fossils and molecular data. Partly it's because different studies use different sampling methods to try and tackle this problem. But a major reason is to do with a very poor fossil record for the earliest chunk of the living lineage's history and evolution. And that's important because even though molecular ages seem to be separate from fossil ages, we actually use those fossil ages as calibration points to kind of, I guess, backbone the molecular data. Most strikingly of all, Polypterids, so this group here on the left, only have a fossil record going back about 100 million years. But as the earliest living group, they must have evolved when the crown node first originated, which we think is about 384 million years ago. So that implies that they have a 250 million year ghost lineage. So where we should expect to see them in the fossil record, but we currently don't. This is a major area of uncertainty, and this is one of the key things that's contributing to that huge discrepancy between the molecular ages and the fossil ages. So I worked with some colleagues in the US and China to re-examine some previously described fossils, in particular this animal here, Fukangichthys, from the early Triassic, about 250 million years ago of China. And we use CT scanning to build up a picture of its internal anatomy. And I've got some pictures on the screen here. So to orient you a bit, this is its nose, the front of its head. This is the top of its head. This is the back of its head, and then this is the bottom. In blue, we have the bones of the cheek and jaws. So you might just about be able to pick out some teeth along here. Purple is the skull roof and the braincase. And then yellow is the gill skeleton, so the bones that support its gill arches. And this work flagged up some quite surprising similarities between these fossils and living polypterids. So here at the bottom is a skeleton of a living polypterid that's kind of coloured, and we've made a 3D model and made it the same colours. I won't go through the anatomy, although I'd happily talk to you for 40 minutes about fossil fish anatomy. I think many of you would probably get bored and not enjoy that. But we can see quite a lot of morphological similarities between the skeletons of this fossil fish, Fukangichthys, and the living polypterid. And these are particularly striking in the braincase, the gill skeleton, and the lower jaw. But it's not enough just to note these similarities. We need to run some phylogenetic analyses to test this relationship. Because we're using fossils, we have to use morphological data, morphological characters. If you remember the example of the fin or the limb, so we do that for lots of different characters. And we can also add some molecular data because we have a number of living animals included in our analysis. And in total, we analysed the relationships of 96 living and fossil fish using 264 morphological characters and 12 nuclear genes. And we found strong evidence across all analyses that Fukangichthys, this fossil fish, is a fossil member of the Polypterid group. So if you remember, we previously thought that Polypterids had this 250 million year fossil ghost lineage where there were no fossils known from the record. And our work has filled in a huge chunk of this. We're able to extend that fossil record back to 250 million years and fill in a huge chunk of that missing record. But this also has implications for fossil fish, for ray-finned fish relationships and for the age of the ray-finned fish crown node. So if you remember the past hypotheses, the oldest fossil we thought was 384 million years old, and the molecular date was anything from kind of 298 million years to 436 million years. But in our analysis, the oldest fossil related to any of the living radiations is 339 million years. So our results suggest that the crown evolved some 50 million years later than we thought from previous analyses. And we see a similar kind of reduction in our molecular dates. So we get an age of 359 million years. So again, this is significantly younger than many previous analyses suggested. But what's really cool is that this reanalysis brings the age of the crown node, so the age of the living radiations, in line with the end of the Devonian and start of the Carboniferous. So this revised phylogenetic matrix, which integrates fossils and living taxa, has the potential to tell us about patterns of ray-finned fish evolution, as well as the timing. And it also means that we can fill in these gaps and we can start to target some of these really important fossils that have been understudied in the last few decades or longer, and try and capture some of the information that they have to tell us that we're currently missing. And this leads into the kind of next phase of my research, which is driven by the broader aim of investigating kind of broader patterns of ray-finned fish diversity and evolution. And it's only possible to answer these questions and think about how ray-finned fishes were affected by things like major mass extinction events once we have a robust hypotheses of relationships. If we don't know how things are related to each other, we can't see how the group was affected by different evolutionary events. So the final thing I want to talk to you about today is some preliminary work which looks at the initial diversification of ray-finned fishes, and in particular how they were affected by major mass extinctions early in their history. So, as well as potentially being the time of the origin of the living radiation, the End-Devonian marks this very major shift in ray-finned fish evolution. So fossil fish in the Devonian tend to be quite boring. They're generally, I guess, fish-shaped. They've got a kind of head at one end, a fairly slim body, and then a kind of standard tail at the other end. They're quite ecologically conservative. They don't seem to be doing anything particularly interesting or innovative. And there are only around 20 to 30 species that we know of in the fossil record. But in the Carboniferous, they're much more diverse and disparate. So there are around 300 known species, an order of magnitude more, and they're much more morphologically and ecologically diverse. So as well as your kind of standard things that look like fish, you also have very deep-bodied animals that are durophagous. So they've got teeth that have kind of either formed into beaks or form a pavement so that they can crush different kind of mollusks and hard-shelled animals. Some of them are very long bodied, so they look superficially like eels. And we've also got a much wider range of sizes, from very, very small things up to very large things. And this diversification really sets the scene for the future domination of ray-finned fishes in the seas. But punctuating these two time periods is one of the big 5 mass extinctions, so the End-Devonian mass extinction, in which around 50% of life went extinct. But the effect of this extinction on ray-finned fishes is really poorly understood. It's variably been suggested that it had no effect on their evolution, that it had a moderate effect, or that it had a major effect. And although we're not really sure yet of the dynamics of what's going on here, partly because we haven't had that stable hypothesis of relationships, it's clear that there's some kind of major diversification of ray-finned fishes going on in the Carboniferous. And we can think about patterns of relationships relative to geological time by doing something called a timescale tree. So we take our underlying a kind of hypothesis of relationships and we scale it to time and look at when different events happened relative to different geological ages. So here we've got the geological column on the bottom of the screen, and then this vertical red line marks the position of the Devonian Carboniferous mass extinction. So when we add our tree, we can see this kind of classic interpretation of ray-finned fish evolution. We seem to have a small number of species in the Devonian, far more species in the Carboniferous, and then perhaps just a single lineage crossing the End-Devonian mass extinction, and then diversifying in the Carboniferous. And this implies that ray-finned fishes were really very hard hit by the End-Devonian mass extinction. So only the single lineage seems to make it through, with all the diversification happening in the Carboniferous. And as hypotheses go, this is quite attractive. It mirrors this narrative of kind of a single plucky survivor making it through this crisis and succeeding in a post-extinction world. And it also shows some similarities with narratives of things like the radiation of mammals after the dinosaurs went extinct at the end of the Cretaceous. But it's not really as simple as that. It's a nice story, but often these things are a lot messier than they appear on the surface. And it's possible that our interpretations are being biased by a lot of missing data. So in particular, we don't know that much about the very kind of latest Devonian fossil fish, those that were alive and evolving right before the extinction happened. So here's one taxon that's historically been overlooked, not least because it's absolutely tiny. So in case you're having a kind of hard time orienting yourself, this kind of grey smear that you can see here on the rock is the body of the fossil. So you might just about be able to make out some scales. And its head is disappearing under this chunk of rock here. And then its tail would have been off the end of the rock. It's not preserved. This fossil has been known about since the early 1900s. And it was described and then essentially put in a drawer and not looked at again. Largely because it's such a small fossil. So this whole block of rock is just a few centimeters long. And this is, again, where CT scanning really comes into its own, because we can take this thing, which looks very unassuming and very unattractive on the outside and also is very small, and we can CT scan it. And we find that actually this is a really cool and really gorgeous fossil fish. So here's its head. You can see this is kind of its eye. So its nose would have been at the front here. This is its upper jaw. You can't really see its teeth because they're so small, but they would have run along here. And this is its lower jaw. And then this is the back of its head over towards the left of the screen. This is a really, really small animal. So this entire head is about the size of your little fingernail. So things such as this would have been really impossible to look at before CT scanning, before we could make these much bigger models. And there's nearly a complete head in here. So we can spin it around and look at it in 3D. We can see we've got bits of the braincase in purple, bits of the gill skeleton in yellow. So there's tons of anatomy in here, which should make it possible to work out exactly what this animal is and what it's related to. And it turns out to have very unusual anatomy for a Devonian fossil fish. So again, I'm going to spare you the gory anatomical details. If anyone wants to talk to me about suborbitals and dermohyles and aortic canals, then let me know and I will talk to you at great length, but I'm assuming most of you don't. But the key message here is that most of the anatomy in this Devonian fish doesn't look like Devonian fossils. It looks far more like Carboniferous fossils. So it doesn't look like we would expect it to look. It looks like things that are much geologically younger. So what is the implication of this for our relationships and hypotheses of how ray-finned fish have been affected by the End-Devonian extinction? What happens when you kind of put these into context? Well, here's our previous hypotheses of relationships in which these very small taxa from the late Devonian are excluded. So we just have this single lineage that seems to cross the End-Devonian mass extinction with diversification happening in the Carboniferous. But when we run a fuller version of this analysis with more of those small late Devonian taxa added in, we get a very different picture. And essentially, what happens is that multiple lineages now seem to cross that End-Devonian mass extinction. So rather than a single lineage making it across and diversifying, it seems as though there's been cryptic diversification happening in the Devonian before the mass extinction. And this has got major ramifications for our understanding of how badly ray-finned fishes were affected by the seemingly catastrophic effect, as well as for rates of extinction and diversification. And there's kind of ongoing work related to this. As I said, this is preliminary. These are preliminary results, which we're still kind of working through. But the underlying message seems to be that our current perception of how ray-finned fish evolution was affected is not necessarily very accurate. And by using CT scanning to add in new details, new anatomical details of previously overlooked fossils, we have the possibility of radically changing our understanding of ray-finned fish evolution. And this has implications for things like patterns of extinction, patterns of evolution, patterns of character change, as well as the age of when different groups evolved. So we've been on a bit of a wild ride through several 100 million years of extinction, covering tens of thousands of animals. So I'm going to leave you with a short summary. Bony fish account for 99% of living vertebrate diversity and ray-finned fishes for 50%. So when we talk about major extinction events and major evolutionary events, it's important to think about the kind of unloved half of bony fish history, not just the things that lived on land and eventually led to ourselves. 3D X-ray imaging has the potential to shed new light onto old problems. So we can not only take new fossils, but we can also take old fossils that we've known about for tens, if not hundreds of years. And we can look at them in a fresh light and get out some of that really important internal anatomy. This allows us to kind of build up these robust hypotheses of relationships that are necessary for understanding macroevolutionary patterns. So using CT scanning and using new kind of analytical techniques, we can build much bigger trees using a much higher number of living and fossil groups, and build up a really detailed picture of how these different things were evolving. And ray-finned fishes have this vast evolutionary history of over 400 million years. They've experienced major mass extinctions multiple times, and through each of these, they've kind of either staggered through or got through quite well and diversified afterwards, and eventually come to account for half of all living vertebrates. So this vast evolutionary history is really perfect for investigating long-term patterns of extinction and recovery in the group. Thank you for listening, and I'm very happy to take any questions.
Thank you so much, Sam. That was really great. I mean, you had some great images there. I really enjoyed hearing all about this area. It's not something that I work on, and it's really inspiring to see this. So if people who are listening do have questions, then please type them into the question and answer box, and we'll try and get through them. And I'll also, if people who are listening on YouTube have questions, then we'll feed those through as well. So our first question from Grace Devine: does the 150 billion ghost lineage of Polypterids show any link with that there are only 12 species?
So it's quite, so the kind of fossil relatives that we see, there are kind of two different groups of them. So the things that we see that are around 250 million years old, there are quite a few of them, but they look very different to the living Polypterids. There are just a few bits of anatomy that are the same, that kind of tell us that they're related. The living group seems to actually have been a lot more diverse in the not too recent past. So nowadays there are only 12 living species, but it seems like over the last 100 million years or so, they are actually a lot more diverse. And today they're restricted to kind of river systems in Africa around the Nile. But in the kind of geological past, they were much more widespread. So they were kind of geographically distributed around the whole world. So including China and Russia and North America. So it seems like over time, they've become more and more restricted and kind of less specios, that there are far fewer species today than there used to be in the past.
Great. So I also had a question just listening to what you were presenting, and this is a more general question you might want to comment on in terms of completeness in the fossil record. So in terms of, you presented those lineages and how they might change when we incorporate more information, but how well have we sampled what might be there, and how does that affect our view of evolution? I mean, this is a broad question, I know, but I think it's important to think about.
So probably not very well at all. If you have a look at, so there's a great resource called the Paleobiology Database. And this is a kind of crowdsourced initiative that the aim of which is to enter all of the fossils that have ever been found around the world for any group. And it's a really cool website, and you can go on it and have a look at where different fossils are known from in different groups. And as you might expect, they're predominantly found in the Northern Hemisphere, and in particular in kind of Europe and North America. And that's because those countries have a kind of, I guess, longer history of more intense research into these areas. So our kind of understanding of evolution is very biased towards these regions. We're missing out on great swathes of the story simply because we either don't have the records, or there isn't as kind of detailed a history of fieldwork or investigations in those areas. So when it comes to ray-finned fishes, we have an even poorer record, mostly because people don't think that fish are very cool, which is very sad because they're actually fantastic and everyone should like working and learning about fossil fish. But there's essentially a peak in the late 1800s, early 1900s, because many of these fossils, particularly in the Carboniferous, are associated with coal deposits. So they would have been kind of quarried out by coal miners and often collected by kind of gentleman collectors and added to museum collections. But then there's been a kind of dip in research history since.
Okay, great. So there are a couple more. Oh,
there's lots of biases, and it's very problematic. There's lots of things that we don't know.
Cool, brilliant. All right, a couple more questions coming in here. One is more of a comment, I think, from Richard Arthur. As a geologist who likes fish and aquatic insects, it's good for his interests. Important to improve our naturalist tend to disregard creatures below the water or the ground. I guess this perhaps tends a little bit and links to some of the comments you're making towards the beginning of your talk about how, in an old-fashioned view, you know, when we look at evolution, we think of us as the peak and everything has come towards that. And, you know, I guess it's important to think about what we consider to be interesting or important.
Yeah, I think if you ask people whether there were more fish or more things that lived on land, they would say there's absolutely more things that live on land, but there are more fish species than there are land-dwelling vertebrates. There are 3 times as many fish as there are birds or mammals, but they don't necessarily, they don't get as much kind of research interest or as much kind of, I guess, general attention, but yeah, there's lots of them.
Brilliant. So we'll move on to the next question from Matthew Status via YouTube. So, are ray-finned fish actively diversifying during the End-Devonian extinction? Is it something about their habitat or lifestyle or niche that gave them an edge over their placoderm counterparts?
Yeah, so it's a really good question. We don't really know the answer, partly because of this kind of lack of research focus. At the moment, we only have around 20 to 30 species of fossils that are known from the Devonian of ray-finned fish. There were obviously more than 20 to 30 species living in the, you know, however many tens of millions of years of the Devonian, but we don't have that many of them. And there are lots of new fossils out there that just haven't been described yet. So hopefully when we have better descriptions of those and we have a better kind of hypothesis of relationships, we'll be able to figure it out. It seems likely that they were actively diversifying. And like Matthew says, it seems like they were probably diversifying into some of those niches that were vacated when placoderms and things like that went extinct. So the kind of classic explanation is that in the early Carboniferous, when placoderms and jawless vertebrates had been kind of wiped out by the extinction, there were lots of new ecospaces, lots of new kind of body shapes and areas of the kind of oceans that ray-finned fish diversified into. So it's likely that that is driving at least some of their kind of radiation in the early Carboniferous.
Right. The next question I've got down here is from Jason Gilchrist, again via YouTube. Can you give us an insight into the difficulty and time involved in studying a single fossil fish specimen? It looks challenging.
Yeah, it takes a long time. So for most of so in an ideal world, you do a CT scan and all of your fossil would be a kind of dark grey colour, and all of your rock would be a light grey colour, and your air would be a black colour. And you just click on the bone and say, and you kind of segment it out in one go. That's typically not how it works. It can take anything from a few days to several months to segment out one of these fossils. So I think the longest one that I've ever worked on, it took about 9 months of every single day kind of physically going through each slice and segmenting out the bones. So it can be really time consuming and very challenging. So it takes a long time, but it's really fun. The CT scanning itself can take anything from half an hour up to, I think the longest I've ever CT scanned anything for was 3 days. Yeah, so that's how long the CT scan takes. And then the segmenting can take, yeah, a couple of days to many, many months.
All right, so we have another question here. This is from Richard Arthur again. So there's a Carboniferous fish in Bude, where it suggested the water was fresh. And do you have any ideas about how to distinguish between fish in these and in saline environments, such as the Plum Marine Basin?
Yeah, so what we can do is we can look at the kind of lithology of the rock. So rocks that are laid down in freshwater settings are different from rocks that are laid down in marine settings. There is a bit of a difficulty when it comes to looking at kind of estuarine or lagoonal sediments. It can sometimes be difficult to work out whether this was a kind of freshwater lake or a saltwater lagoon or some kind of, there may have been an interplay between the two. So it can be really difficult to figure out these kind of estuarine habitats. What we can often do is have a look at kind of associated fauna. So there are some animals that can only live in freshwater environments and some animals that can only live in saltwater environments. So we can have a look to see whether that kind of gives you any clue as to whether this is a lake or a saline basin. With regard to the specific question of Bude, there's a really cool animal called Elenichthys that is known from the rocks in Bude. So I have a specimen of Elenichthys from Bude, which I've CT scanned and will be working up at some point when I have access to my kind of specimens and CT scanning equipment again. So if you watch this space, I can hopefully tell you more about that specific example. In terms of cost, it again varies. It can be anything from, I guess, £100-ish would be the average, but it can be less or it can be more depending on the size of the specimen and how long you're going to scan it for.
And I have one other question related to that, just in terms of a follow-up, which is where do you think this technique in CT scanning is going to be going in the future? What would you say is the research frontier?
So having the ideal world would be to have some kind of automatic segmentation where it's not me sitting in a room for 9 months kind of individually picking things out. At the moment, it seems like there's machine learning options for some kinds of CT scans. So if you're looking at kind of teeth or scales or something very small and very regular with a uniform matrix, there are some ways to kind of automatically segment that out. For most of my specimens, it's just not possible because the contrast or the difference in X-ray attenuation between the fossil and the rock just isn't good enough. And also the fossil is so variable in anatomy. It's not just one tube. It's kind of, you've got a big bit here and another small bone here. And so I think that would be very nice, but I don't think is anything that's kind of on the horizon anytime soon. Also having kind of more rapid access to CT scanning. You often don't know whether a chunk of rock contains a fossil or not. And the only way to find out is to CT scan it. And if there's nothing inside it, that can be a very expensive and time-consuming way to find out that you don't actually have a fossil in there. So it would be nice to have some kind of, I guess, way of looking at these things very quickly to determine if it's worth doing a proper CT scan or not.
Yes, brilliant. Excellent. Well, I think we have now actually, we've gone through all of the questions that we have, and we've actually taken up almost an hour. So if there are no further questions, I'm going to bring this to a close. I would like to thank Sam Giles again for her talk. Really interesting, super inspirational. I mean, it's a long time since I've thought about fish in this way, and I really enjoyed what you had to say. It's been a pleasure, and thank you so much for taking the time to come and give this public lecture. So, and I want to thank everyone who has tuned in to listen. It's really great to be part of this community, and we will be having further public lectures coming up. So please check out the schedule online on our website at the Geological Society. So thank you very much, everyone. It's been a pleasure.
Thank you very much for having me. All right, bye.